Hello, greetings all, and good afternoon. My name is Naomi Chery, and I attend to New York City College of Technology, and my major is in biomedical informatics, and my ASRC initiative is in structural biology, and I will be focusing on the study of ryanodine receptor structure function through their course of evolution. Well, I know you're wondering, what is a ryanodine receptor? Well, they're just simply proteins. They're composed of three different isoforms, RYR1, 2, and 3. Their main function is to mediate the release of calcium ions. They are responsible for the excitation contraction throughout the muscles of the body. We primarily find RIR1 throughout the skeletal muscles and the smooth muscles, example, the esophagus. RIR2 can also predominantly be found in the cardiac muscles. Also, RIR3 is predominantly found in the brain and diaphragm. These receptors are the largest identified types of ion channels, as you see displayed here. IP3R, also known as inositol triphosphate, is also a protein. They are also found and composed of three isoforms, IP3R1, 2, and 3. Functions of these receptors mediates also calcium ions. They serve as messengers in the hormones and also is responsible for muscle excitation contraction. The locations of these receptors are widely expressed throughout the nervous system and intracellular organelles. The structure is also half the size of the ryanodine receptor structure. At this point now you're wondering, what is the significance of this study? Well, now that you have a general overview of RYRs and IP3Rs, you can make a decision on what happens if any mutations happen to these calcium ion channels. Their protein channels can be linked to many human diseases if there are mutations that are occurring. It can be motor movement, cardiac disease, developmental disability, and traits and conditions. RYR2 mutations can lead to ARVD, which is very, very poor dilated and constriction of the ventricles of the heart. A recent study that examined coevolution of these two channels suggests that RYR arose from ancestral IP3-like channels. By incorporating these domains, the sequence identity shared between the two proteins are only approximately 17%, whereas if you see illustrated, the transmembrane region increases to 35% sequence identity. As you can see, here are the results of each protein when you look them up in the protein data bank. Each receptor has their own unique PDB code, and here it will allow us to get the UniProt codes that will allow us to download the sequences in a FASTA format. Now that we have generated our sequences in the correct format, it is time to blast. This will allow us to upload our sequences in a database that calculates its statistical significance. We just simply paste our FASTA sequences that we derived from our PDB protein databank into the query, and then we change the algorithm parameters and set the maximum targeted sequence to 10,000. Yes, we are trying to search for 10,000 sequences. After my computer crashed about 10,000 times, we finally derived our statistical 10,000 sequence identity hits on blast. Now it is time to save it. We save it in a text edit file as you see illustrated here. One sequence is about 5,000 residues in length, and we have 10,000 sequences. Once we are finished blasting all RYR isoforms, including IP3, we are ready to clean these sequences using CD hit. This will allow us to curate our sequences. First, we choose the file that we would like to upload. Then we take the sequence identity cutoff to 95%. Then we take the sequence length skip to 2,000 residues or less. Now that we have our cleaned, curated sequences, it is time to upload it on the Mega software. This is a molecular evolutionary genetics analysis software tool that is used to consensus sequences from alignments and also construct phylogenetic trees. There are different methods on how you would like your multiple sequence alignment to be constructed. In this case, we used muscle. There are other methods such as like CLUSTAL-W, for example, but I just wanted to illustrate on how the sequences are aligned before and how they are aligned after using the muscle domain. As you can see vertically, the amino acids are very much so aligned in comparison to the before picture, where the amino acids are viewed as a bit more scattered. The research is still ongoing. The results are to be continued as we are trying to derive a consensus sequence that will later on be used to construct our phylogeny tree. So stay tuned. I would like to take this time to give a special thank you to the organization that contributed fundings to the CUNY Summer Research Undergraduate Research Program. This opportunity provided many resources to minority students such as myself interested in the research science field. This program will propel the agenda in redefining diversities in areas of science while promoting inclusivity. So thank you guys for this opportunity. Really appreciate it.